Yes, thank you to the panelists. Thank you to the speakers. I just wanted to perhaps draw some insights together, particularly on the panelist discussions at the end. But it's been a good discussion and dialogue between, as well, the interface now between what we're doing in the protein and AI ML engineering space. I think we have made headway in understanding what are the questions biologics engineers have, and that, can we do LO? The answer is clearly yes. For a range of attributes, can we do lead generation, de novo, be a B cell computationally? It looks like it's possible, and a few groups are doing it. From an AI ML or deep learner's point of view, do we have fair enough data? Yes and no, but let's come back to that. Particularly, I want to follow up on what Sandeep was saying and Victor. Which de novo strategy for lead generation is best? We heard lovely descriptions of fragment-based discovery from Pietro, some structure-based, sometimes called repertoire engineering from Biologic Design, and Sanofi had a nice paper on that and discussed it at PEGS. We didn't really hear anything or so much on the kind of more complex diffusion dynamics orientations that we see published from Stanford and Baker. And we have yet to answer the business question about which of these solutions is actually most appropriate, which one is going to deliver benefit for patients, which are cost effective, which are sustainable. That's perhaps a question we're not quite ready to answer yet, but we will definitely head towards it. But just to conclude on this question of data, particularly, have we got data that's fair enough? I think one of the things we should follow up on today as a community of engineers from different disciplines is who is interested in collaborating in this space. As I think Sumi mentioned earlier, we can learn a lot from the chemistry world because they have been working in the machine learning space for much longer than we have as biologics. And they actually have two good paradigms for sharing data. One is called Melody and the other is called Cache. I want to propose that if you're out there and interested in sharing data as an individual or potential or as a big company, I'm saying this as Andrew Buchanan, but if you're interested in sharing data in a secure and federated manner to build a better, let's say, supermodel, and I think this would potentially work in the developability space, mirroring something called Melody, I would definitely be interested, and other peers and other companies would definitely be interested, how we could enable what is essentially a secure, federated learning with a trusted third party. We talked a little bit about as well, how do you benchmark computational models? That's an open challenge, and you see that in the peer-reviewed literature. So I think it's been a very interesting set of talks, a very interesting discussion. I think we can collaboratively make headway in data sharing, particularly in developability. So let's try and work out a way to actually make that happen. If you're interested in continuing the discussion with machine learning and biologics broadly, I would really recommend two other channels to follow. One's called ML for Protein Engineering, a seminar series, and the other is called, will be a half day called Marble, again, AstraZeneca sponsoring with the ECML-PKDD. I'll try and send out the links later. But those are two good events that are on, well, one's an event, the other's a seminar series that happen on this topic. But to conclude, finally, I suppose a last thanks to all the speakers and the panelists. Thank you for engaging as attendees and posting questions to challenge the speakers and panelists a little bit harder, which is great. So thanks for that engagement. And again, thank you to the Antibody Society for hosting, particularly to Janice for enabling this day to happen over what started as just a question. Could we do it? No, we've actually done it. So everyone will get back to their data, but let's not lose contact around how we can collaborate and share data, particularly in the developability bracket. So thank you very much for today. I think at that point, we'll close the symposium, and thank you again for attending.